We are at chapter 18.3 and we are talking about microRNAs and small interfering RNAs. So let's talk about how those are made and what they do. So we found that only about 1.5% of the human genome is used to code for proteins. However, 75% of the genome is transcribed in a cell. So if we're transcribing all of this DNA into RNA, how come so little of it is being translated into proteins? And so a couple of answers that we have is that a small fraction of the non-coding DNA consists of genes that code for other RNAs. So your transfer RNAs, your ribosomal RNAs. Those are your ribozymes. Those are your enzymes that are RNA. We also have introns are transcribed but not translated. So they are transcribed into RNA, but they are taken out before they are turned into proteins. And so research is showing us that there's a significant amount of the genome that is coded into what we're calling ncRNAs, or non-coding RNAs. And these are essentially strands of RNA that have a job in the body, and not necessarily enzymatic, just a job. They do jobs around the body. So there's a diverse population of RNA molecules that participate in regulating gene expression. So we're going to talk about how these non-coding RNAs can silence genes. And there's two molecules that are used to silence genes, and those are microRNA and small interfering RNA. So these are two examples of non-coding RNA. And the pathway in which they're made is called the Rna interference. So here is the RNAi, or RNA interference mechanism. And we call it the Rna interference because it is going to interfere with gene expression. It's going to silence gene expression. So let's talk about the pathway in which the small interfering RNAs and microRNAs are made. Right here, we're going to start off with a double strand, a long double strand of RNA. And it's going to be out of the nucleus, and it's going to attach to an enzyme called dicer. And dicer is going to dice it up, and it's going to cut it into short-stranded, double-stranded RNA molecules. And so the short double-stranded molecules are going to attach to another enzyme called argonaute. You don't need to know that. But argonaute is going to then choose one side of the RNA molecule to bind to. And then the other side is going to be cleaved off and disappear. Not disappear, but it's going to be broken apart, degraded. Now, it is this argonaute and the small RNA molecule, single-stranded RNA molecule, they're going to connect to some other proteins to make a complex. Complex is called RISC, but you don't need to know that. And RISC is going to match up onto the RNA to silence it in a couple of different ways. So there are two different RISC complexes. There is the small interfering RNA complex, and so it's going to use its nucleotides on the small RNA strand to base pair exactly with an exact match. So it's going to be a very precise tool. It's going to line up, it's going to look for a very specific nucleotide sequence to line up to, and then it's going to cleave it. And then it's going to degrade the RNA that has been cleaved away so that the gene is silenced. But it is looking for a very specific spot. It is looking to pair exactly. So every nucleotide has to match. Our less specific tool is going to be our microRNA complex. So still a complex of proteins, and it's going to have the single short strand of RNA. However, it's going to be able to loosely match, meaning it's going to match with certain nucleotides that aren't necessarily an exact match or are correctly matched. So it'll be a looser bond to the mRNA, However, it will still be able to degrade or silence the gene. It might either block translation, might attach to it and block it from translation, or similar to the siRNA, it'll degrade it. It'll cleave it and degrade it. But this is your less specific tool. This is your very precise tool. Here we go. So recently discovered there are approximately 1,500 genes for microRNA. So this is new information, and it's a growing field of research. And it regulates about half of all human genes, so a huge player in gene expression. This is a much bigger player in gene expression than we had ever thought. We used to say that proteins did all of the jobs pretty much. However, we're finding that these microRNAs play a significant role. So scientists use microRNAs and the small interfering RNAs to disable specific genes. They silence those genes in the lab in order to investigate their function. So if you turn off a gene, you can see what it does because it's not doing it anymore. So this has been used as potential cancer therapy, and it appears to be very promising in cancer research. So scientists believe that this silencing effect evolved as a defense against viruses, because there are some viruses out there that have this long double-stranded RNA. And so we believe that the ability of the cell to defend itself against viruses and to break that DNA down from the virus has evolved into this very helpful tool. So in addition to regulating RNA or our messenger RNA, non-coding RNAs also regulate transcription by remodeling DNA, changing the form. So DNA replication occurs in S phase, and so at that point we need to condense it so that it can replicate. And then at mitosis, it needs to be unwound. It needs to decondense or uncoil during S phase. And then mitosis, it needs to coil up so that it is organized for cell division. So in yeast cells, scientists have found that the siRNAs, or the small interfering RNAs, work with other non-coding RNAs and enzymes to condense the chromatin. So it is working to help change the form, or to coil and decoil, that's the word, the chromatin. And so small interfering RNAs generally do not occur in mammalian cells. However, we believe that there are other non-coding RNAs involved in this process. So there are long non-coding RNAs as well. So we don't just have these short ones, we have the long ones too. So ranging from 200 nucleotides to thousands of nucleotides. The job of one long coding RNA strand is to work in the female X chromosome inactivation. So it's pretty cool. The gene that is inscribed in the DNA for the long coding RNA strand is transcribed from the X chromosome that is actually going to turn it off. So it is going to be transcribed from the X chromosome, and then it's going to essentially methylate and turn that X chromosome off, which is pretty interesting. So scientists believe that long coding RNA play a role in sort of moving chromosomes around, bringing them together so that they can function together, and they also act as a scaffold for DNA. So they just have tons of these little jobs all over the place, making things happen. So there are many levels of regulation in gene expression, and this is what allows for the complexity of life. We have the A's, T's, C's, and G's in DNA, but so many different combinations that allow for almost infinite possibilities. And so as life continues to increase in complexity, it begs a question. Are these non-coding RNAs, these microRNAs, and these small interfering RNAs, and these long non-coding RNAs, are they a key player in this process of creating complexity? Okay, I hope you guys have a great day, and we are done chapter 18.